want to say again, because of all these changes, there's been a mistake in the Moodle that according to the Moodle, I was supposed to give 2 immunology lectures today, but that wasn't correct because I'm going to give you the last of my lectures to you. This is on virology, and the next lecture I'll give is an immunology lecture on vaccines. So now this being the last microbiology lecture with me anyway, this is more of a general discussion but on this really serious matter of emerging viruses. And the reason why it's important, because as clinical health workers later, you are part of the community, and you have to be aware of what is going on around in the community. This COVID-19 crisis at the moment, you're privileged to live through a pandemic where you'll be able to tell your children and grandchildren exactly what it's like. Because it shows up that despite all of our advanced knowledge of medical science, we're still helpless when it comes to something like these viruses. We have no defenses other than containment, social distancing, quarantine. Because these things are really, really difficult because they can multiply so fast and they can change so much. So our only way of defending ourselves against these things is by boosting our immune system. But even here with COVID-19, it's suggesting that our immune defenses aren't 100% effective against something like this virus, where data shows that we only have protective immunity for up to 6 months. Now, what is this? Now, bearing in mind, as I said, that this lecture is essentially more of a seminar or a discussion time, you're going to ask, well, what must I study for this lecture? What you need to know is the names of these viruses as we go through this lecture that I described, and just know the general principles of these things. So, for instance, if I talk about influenza, obviously you know what flu is like. But what is important is when we talk about influenza epidemics or pandemics, you're constantly reading from the WHO or whatever that there's an H1N1 or H1N5. What is H1N1, right? So just these general properties of these various organisms. You will hear more about these individual viruses next year when we go into more detail in the multi-system type blocks. So the objectives of this lecture is to talk about this danger to world health with regard to emerging and novel viruses, and the causes for their appearance. Namely, one, that these viruses, particularly RNA viruses, are able to adapt to the immune system, as well as to, in terms of their mutation, that they can evade our immune defenses. But the other reason why these viruses are a danger to communities is because of human activity itself. That the higher our population increases, the more we encroach into areas where we shouldn't be in the first place, where viruses have adapted to live with their hosts, namely wild animals in tropical rainforests and so on. When we encroach into these areas, we create more risk for these viruses to jump from their natural habitat into the humans, so that when we get back to our cities, we then pass this virus on between ourselves and create these pandemics. The other reason in human activity is the ease of travel. With jet travel now, with political disruptions in society in terms of wars, poverty, you get huge migrations of refugees going from one area to another. Many of these communities where the virus is adapted within these populations, when they migrate to another area, they can allow for conditions to pass this virus on to these virgin territories to start infecting again. And then finally, to talk about world changes in terms of climate change, big climate disasters like hurricanes, earthquakes, where society is disrupted. And that breaks down all of the infrastructure in terms of clean water, monitored food, and so on, that allows for pathogens, not only viruses, to then spread in the community. So what do we mean by emerging viruses and re-emerging viruses? So emerging viruses are viruses which we haven't previously encountered, which is not a particularly well known to various parts of the world. And again, through major changes in say, air travel, you can then get the migration of viruses which previously were endemic in, say, East Asia, suddenly appearing in North America. Re-emerging viruses refers to the fact that certain viruses like measles, mumps, rubella, where vaccines for these were already implemented in the 60s to create this, what you'll learn about in the next lecture, the situation called herd immunity, where with enough people who are immune to a particular virus, we can deprive the virus of more food or more people to infect, so that essentially we can wipe out this virus. The biggest success story for this was smallpox. When the smallpox vaccine was developed in the 60s, the WHO embarked on a huge worldwide campaign to make sure that everybody in the world was vaccinated. And as a result, this virus has been essentially eradicated. We've been having problems trying to do the same with polio and measles because of politics and because of economics. The fact that particularly in Western countries with access to Google, Wikipedia, a lot of people think they're better than qualified scientists to make judgments. And so you have this anti-vaccine movement, which is significant in breaking down this herd immunity. And as a result, you have situations like, I think just 2 years ago in Disneyland in the US, there was a big outbreak of measles virus due to some families going in there, not getting their kids vaccinated, and then spreading the virus around. And you're getting pockets of measles even in the UK also. So that's what's referred to as a re-emerging virus. So as I said, one of the reasons why these viruses can break out or escape, particularly with RNA viruses, is because of their high mutation rate. And this mutation or this variability in the viral genome can come in three forms. One is the fact that, as I said, RNA, the mechanism of RNA replication is a very error-prone process. It doesn't have as strong proofreading ability as DNA replication. So you get a lot of mistakes, inserted, misread bases put into the new RNA genomes as each replication cycle happens. That is one part. The other part is that viruses like retroviruses, because they can, the enzyme reverse transcriptase can switch from RNA into DNA, that also introduces a lot more errors. With this insertion of the this proviral DNA into the host cell DNA, when that that viral DNA comes out of the host cell DNA again, that itself is not a precise enzymatic process. And what essentially what it does then is it can take bits of human or eukaryotic DNA out with it into the viral genome. And so incorporate host sequences into the virus particles. And this is particularly what we see with DNA viruses like the herpes viruses, which their major property is latency, so they can insert themselves into human genome. So you find a lot of herpes viruses have human genes in them. Genes like cytokine receptors, genes like proteins which can disrupt TAP enzymes so that when they infect a cell, they can block HLA expression on cells. So the bottom line is that there's a constant warfare between these invading viruses and an immune system to defend our bodies. And with each time the virus wins or the immune system wins, very, very similar to what I told you about with the development of tumor cells. So expanding on this, these routes of variation, particularly with these RNA viruses, we can divide them into recombination. So you know from basic biology that DNA, this is how we get evolution, because during meiosis, you get recombination between the diploid chromosomes, right? So paternal chromosome can cut pieces off and then join up with the maternal chromosome. This is how you get adaptation. The same thing can happen with RNA viruses. If you have two different RNA strains infecting the same cell, during replication of the new viral RNA genome or the messenger RNA, what the polymerase enzyme of these viruses can do is they can read part of one strain and then jump over and read the other strain. So essentially you get then a hybrid genome in the progeny viral. So this is recombination. And this is what I meant, if you remember from the first lecture, about ambisense RNA. So ambi refers to this ability to read part of one type of genome and then read the information from the next part to create this hybrid progeny. That also related to this template switching as well, which is the same sort of process. This one with a non-replicative is actually a physical break between the genomic strains. So if you have this, I'm colorblind, is that green or red? That's green. So basically you have a segment of the parental RNA cutting off and then joining up with this blue strain. And then that, when that gets read into new RNA species or messenger RNAs, you get these hybrids. This one is referred to as this ambisense, where the polymerase can read part of it and then jump and then read the other part. Transduction refers to the fact, what I was saying, where virus genomes which can insert into the host, and then when they reactivate and come out again, they can take parts of the host with them. But backwards from this, we also now find that viruses can also leave parts of their genome in the host sequence as well. And this, a lot of studies have been made on this, particularly with mice, where we now know that every mouse that comes out in the wild, you can see virus sequences in them from millions and millions of years ago. These are called endogenous viruses. And with gene sequencing, we are now detecting more and more of these ancient viral sequences in our human genomes. Most of these sequences are what we call junk DNA, where they don't actually code for anything. But nevertheless, we are now thinking that some of these sequences at least can contribute to our evolution and even our behavior. Recently, it's been these endogenous viral sequences are associated with this very closely studied but very little known disease. I don't know if you've ever heard of it, called chronic fatigue syndrome. Chronic fatigue syndrome is a metabolic state in a small population of people who actually have no energy at all. And doctors have studied this for 30 years, but we still don't understand whether these people are just hypochondriacs and just putting it on, or whether they actually have a true physiological defect in their metabolism, but they just have no energy. And now it's considered that some of these endogenous sequences might be playing a role in this. And then finally, there's this important process, which refers to that process of what I told you about, genetic shift. And this is in reference to these viruses like influenza, which have multi-segmented genomes, like chromosomes. So that if you get two different strains infecting the same cell, you can get reassortment or mixing and matching of these components to create hybrid progeny. And this, why we call this a genetic shift, is that it's a major jump in the actual phenotype of these progeny. So that if they infect a host, that host never seen that antigenic pattern before and can overreact and create something like a cytokine storm which can be lethal. The 6 major new, what we there's 1, 2, 3, 4, 5 what we call new viruses, and the one emerging, re-emerging. The 5 new viruses, well, 4 of them are essentially what we call arboviruses or hemorrhagic fever viruses. These are traditionally restricted to tropical rainforests, tropical areas. And because of that, they are part of what are called zoonoses. Have you heard of the term zoonosis before? So zoonosis is just like you learned in parasitology, like with malaria. These are pathogens which use different vectors or different species in their life cycle. So just like the malaria parasite goes through the mosquito life cycle and then into the humans, these viruses use an insect vector to go into an animal vector, which can then pass into a human vector. So they involve multiple species. Yeah, so these viruses can pass into a mosquito. The mosquito can feed on birds, feed on horses, and as they are taking blood or passing the virus into, say, a bird, the bird can replicate the virus, which can pass into its blood, so they get viremia. Another virus can feed on that bird and then go and feed on a horse, and then pass the virus into the horse. And then from there, either with people who work with horses, or if a virus feeds on the horse and then feeds into a human, the virus passes it on again through there. So in a recent study, in the latest study by a WHO group in 2012, it's been concluded that 75% of all emerging virus infections are zoonoses. Of these major ones, there is the Zika virus, Ebola, influenza, which is not essentially a tropical arbovirus, but it's a particular problem. Norovirus, which isn't a tropical virus, is associated with masses of people in isolated cases, like I said in the last lecture, on cruise liners, military establishments, schools, where there's a communal food source. If there's any contamination in the source, it can spread. And then this virus called chikungunya, which originated in West Africa. A lot of these very dangerous viruses like Ebola, chikungunya, Marburg virus originate in West Africa, in the rainforest, because these are remote, and those viruses have managed to live happily with their environment, with animals like monkeys, gorillas, chimpanzees, and not cause disease. HIV is a particular case in point, that it originated in West Africa in monkeys. Now, at this point, I should talk about something like Ebola. You've all read the news. You know how deadly dangerous Ebola is. And like I said, it's got a very low infectious, low infection, that you only need one virus particle to get into your blood to kill you, right? But the thing is that a virus like Ebola is a very inefficient virus because it's like a wildfire. It's like a forest fire. When these viruses burn through a community and have a high mortality rate, basically what they're doing is killing off any more fuel for the virus to survive. So by wiping out a whole community, the virus also dies. The most successful viruses are viruses like herpes viruses, which have learned to adapt and survive with their hosts so that they can stay in the host for the rest of its lifetime. So Zika virus is a particularly good story in terms of how a virus spreads with human activity. I don't know if any of you know about Zika virus or you followed the news about Zika virus. This only happened 4 years ago. So basically, Zika virus was reasonably well known in Asia, East Asia. Places like Vietnam, Thailand, and so on. And then it spread to the Pacific Islands near New Zealand. Places like Tonga, Fiji, and so on. It caused a mild fever in these areas. This was around 2007, 2008. Then the Olympic Games happened in Brazil, and athletes from these areas went to Brazil. And a few months after the end of the Olympic Games, you started to see more and more kids in Brazil being born with microcephaly, where they had really small brains, small heads. And when they looked at these kids, they found this virus which originated in these Pacific Islands, the Zika virus. And it's still a problem now. And this virus is spread by mosquitoes. So the idea is that these athletes went to Brazil carrying virus in their blood, got bitten by mosquitoes in Brazil, which then passed that virus on to local populations. Measles, as I said, is a result of human ignorance of the anti-vaccine movement. Again, I don't know if you've heard of this guy, Andrew Wakefield. This was, when was it? 1999. There was I think he was a pediatrician in the UK who published a paper that suggested that the measles, mumps, rubella vaccine caused autism in babies. And this was published in the Lancet. And it got into the newspapers. And so the general public, particularly mothers who had babies, bought this and said, we're not going to vaccinate our kids with measles, mumps, and rubella. And this took on, this idea got caught on such that so many unvaccinated babies came up with measles, mumps, and rubella since then. His paper was later retracted, but by then the damage had been done. And this movement spread in the US, particularly with high-profile actresses who were more concerned with their kids. So the problem with science is nothing is absolute. We can't say with 100% certainty that anyone who gets a vaccine isn't going to get an adverse effect. But science is based on statistics, and we base decisions on relative risk assessment. So general public don't understand this, particularly mothers who concerned about their little Peter or whatever, and so make these crazy decisions without considering the rest of the community. A particular problem in emerging or re-emerging viruses is the influenza virus. And just in the past month, there have been thousands of chickens, ducks, and turkeys being culled in the UK, Europe. With the COVID-19, thousands of minks in mink farms have been culled. With these birds, the culling is due to the detection of this particularly worrying strain of influenza, H5N1, which has a high mortality. Now, the reason why these birds are always targeted and so closely monitored is because influenza, as I said, is a zoonotic pathogen. So it can spread from migratory wild birds to these poultry farms and then pass that virus on into these domestic chickens and whatever, and then pass that on to humans. So every year, the WHO wildlife authorities are constantly monitoring migratory ducks, birds, and whatever. And if they see mass deaths in particular areas, they take samples of these animals to look at if there's a particular virus in them. So they are particularly cause of concern. What do we mean by H1N1, H5N1? Basically, all you need to know is that the influenza virus infects or binds to a target cell through this receptor called H, or for hemagglutinin. So that is the receptor. So the type of receptor H1, 2, 3, or 5 is how the virus gets in. Certain strains with particular hemagglutinin antigens, particularly H1N1, which caused the Spanish flu in 1918, and H5N1, which caused the big outbreak in Spain, in Mexico in 2009. These are the ones seem to be highly virulent with a high mortality rate. So we constantly on the lookout for that. The other antigen, neuraminidase, is the enzyme that the virus uses to get out of the cell once it's replicated. And we've found through research that people who have antibodies to this neuraminidase can block the exit of these new virus particles out of the cells. So that's why we are constantly looking at the strains of H and N with each new season of viruses. And as Dr. Asif will tell you in his next lecture, is that every year when they bring out the new flu vaccine, there are bodies which monitor, just like weather reporting. They try to predict what new strains of influenza will be hitting Saudi Arabia or any parts of the world based on the epidemiology that they're seeing, and then try to design vaccines to cover for the next season, sort of thing. Influenza is part of this group of respiratory viruses, which are a particular problem in the community. We know now about the coronaviruses. The first one was SARS, which stands for Severe Acute Respiratory Syndrome. Saudi Arabia, we know about Middle Eastern Respiratory Syndrome. And now we have the SARS-2-CoV. SARS-2-CoV is closely related to SARS-1. MERS is luckily didn't kill as many people, but it is much more virulent than SARS-2-CoV. The thing about coronaviruses is that we've known about coronaviruses ever since the 50s. And with every winter, a large percentage of people who got common colds or flu, who weren't sick enough even not to go to work, were infected with coronavirus. We didn't even bother to look at what virus they had. We just assumed it was a flu virus or a cold virus. Many of these cases could well have been coronavirus. Why the coronavirus since SARS has suddenly become so virulent, so infectious, we don't know. We think, again, this may be due to some evolutionary adaptation of this RNA virus, or that it may have jumped from an animal species into human hosts. There's another relatively new virus called metapneumovirus, which was first discovered about 6 years ago in Japan, which was particularly serious for in kindergartens and daycare centers, where kids were particularly hit by this virus. And so again, this is being closely monitored every year to see that it doesn't spread worldwide. H5N1 is this one that I've just talked about. With regard to these arboviruses, as I said, we have chikungunya, dengue, and Zika virus. Dengue, which you'll hear about next year, is particularly prevalent in Asia. So if you go to places like Singapore or the Philippines, when you leave the airport, you'll see signs warning you of constantly monitoring to make sure that you don't leave, like in your apartments, you don't leave pools of water around with your plot plants or whatever, which would allow for places for mosquitoes to breed. So it's a particular concern here. The thing about virus like dengue, and which we also know have seen with COVID-19, is this process called antibody associated infection. Basically, what this means is that opsonization, as a result of antibodies binding to the virus, does not neutralize the virus, but actually amplifies infection. And this is because these viruses like dengue can infect macrophages. So macrophages harming Fc receptors for IgG, you get IgG binding, so-called neutralizing antibodies binding to the virus, but then attaching onto macrophages via the Fc receptor, allowing these viruses to infect the macrophage. And so the macrophage can then pass through the body and so increase the severity of the infection. West Nile virus is an example of human activity that has spread this restricted virus. So West Nile virus before 1999 was particularly well known in the Middle East, in Egypt, in Israel, in this part of the world. It causes encephalitis. Not serious enough to kill you, but serious enough to lay you out in hospital in ICU for a while. Prior to 1999, it was never known, never seen in the West, in America or Europe. Then in 1999, cases were seen in New York, very few cases. In 2002, it is now spread through the whole of the US. Zoologists try to figure out how this virus appeared in New York all the way from the Middle East. What the current theory is that with flights coming from the Middle East to America, you had virus-containing mosquitoes that managed to stay inside the wheel compartments of these airplanes. So that when they landed in New York, these mosquitoes got out. They then infected horses nearby in the zoos and in horse farms, and then passed it on to humans. Other viruses, well, no, not so much horses, but birds. And so over the months and years, you had this cycle of infected mosquitoes where the virus was replicating, feeding on birds, passing the virus in the birds. The birds are harboring replicating virus, and you have the cycle between the bird and the mosquito. In the course of this, the infected mosquitoes then infect horses and places like stables and horse farms where workers are working close to these animals, breathing in the dust or the dung that the horses excrete. They then breathe in the virus and then take it in. So this is an example of a zoonosis. But an important point to note is that the horse and the human in this kind of cycle are known as dead-end hosts, because these hosts, the virus does not undergo viremia. It just causes disease. So it doesn't replicate as significantly within these hosts. So even if a mosquito feeds on these infected individuals, it doesn't carry enough virus to then pass on into the host. So this cycle between a bird and a mosquito is where you're getting continuous amplification of this virus. Bats are a particular problem, and what we strongly suspect bats as being the reservoir for MERS as well as COVID-19. And if many zoologists have looked at bat colonies and find many pathogenic viruses in them. but they don't seem to kill the bats. And so a lot of research has been going into looking at the immune system of these bats. And what it's found is that they seem to have a high level of tolerance, that they do not, whether they have strong regulatory T cells, but they do not make significant amounts of inflammatory cytokines against these viruses. So they're one of these perfect adaptations to these pathogenic organisms. Because remember, I said that many of the symptoms that you get as a result of virus infection is due to your immune response, due to the inflammatory cytokines you make. So these bats are ideal carriers for these pathogens. And then when they fly into communities and drop their feces, or sometimes you get bats that feed on the blood of other animals, they can pass on these pathogens.
Doctor, about the COVID-19, it's just respiratory, droplets transmission, right? It's not by fecal-borne transmission.
It's by respiratory, yeah. But the thing is it respiratory between, so one of the ideas is through respiratory, through feces or whatever, it gets into a secondary host, right? Now in the case of MERS, the idea is it started in the bat, got to camels. According to Dr. Arthur, the latest studies show that certain Bedouins, they live with their camels. They have the camels in the houses. So when you are living in such close contact with animals, you can have a higher chance of getting the virus jumping through breathing in whatever excretions that the animals do. And this is the problem with the COVID-19 in China, was the idea that because the farming practices of in areas like China, they have such concentration of live animals, and particularly in these farms where the people are living close to all of these animals, as well as in the markets where they have these live animals in cages where customers are walking around exposed to these. There's a high risk. The idea is that there's a high risk of the virus jumping from these infected live animals into the humans.
And I heard in China they have something that's called the wet market.
Yeah, so the wet market is what I mean, because for the Chinese culture, they want their food as fresh as possible.
Yeah, but they are eating in that, and that's what it
It's not that they're eating, but they're keeping the line. So here in the West, we all used to getting our meat in prepackaged, skinned, processed packages. Whereas over there, they like to take home live chickens and then chop their heads off and cook them straight away. Right? The other problem too is with these viruses like Ebola, is in Africa, because these people are so poor, right, and there's no supermarkets, or the tribes that live, the villages that live in close to rainforests, they go into the rainforest to hunt for live animals, like monkeys, like deer or whatever, and then take them home to eat. This is what's called bushmeat. And this is one of the biggest sources of viruses like Ebola. So again, this is human activity that's creating this risk. So the bats, by harboring these pathogenic viruses, are a serious risk for passing into communities. These two viruses, Hendra and Nipah. Hendra was a particular problem in Australia in the late 90s, where it broke out in a horse farm in some stables. The stable workers who were working close to the horses breathed in the virus, which got it by breathing in feces excreted by bats, infected bats. And so, as I said, as I just referred, the human activity also plays a big role in spreading these emerging viruses through mass migration, through travel, through increasing density of living or encroaching into these rainforests and then exposing themselves to these viruses, through farming practices of concentrated chicken farms, duck farms, and then living close to them, breathing in any potential pathogens, through global commerce, spreading or growing meat, having chicken, exporting chicken from one part of the world and sending off to the other part. This was again found in COVID-19, where soon after the outbreak, Chinese authorities were detecting COVID virus from fish that they'd imported from Norway and so on. Social behavior. This is a particular problem for you as medical workers. So you have a danger of needle stick injuries. But also there was a case about 15 years ago of a dentist in Florida who had hepatitis C and passed the virus on to something like 25 patients, because that virus is spread by blood. So when you're doing dental procedures, if the person's got any abrasions in the hands, you can pass the virus on. So as Dr. Atef will tell you in the next lecture, this is why infection control is so important. And this is why you need to be very aware of this. In terms of recreational drugs, STDs, that's pretty obvious. HIV was a particularly prevalent concern. So it was too young to been lived through that, but this is why it created such a big problem in Africa and Southeast Asia because of sex tourism. A lot of prostitutes were passing this virus on to all the customers. Okay, that's it. So we'll take a break and then we'll start again at 10 past.